So my name is Eric Castillo. I'm with the Tier Dresden. So welcome to the first session of the HiPEAC conference. We'll have the papers in this session. So the first paper, as you can see, is going to be presented by Can Feltino. So he's a PhD student at the Safari Research Group at ETH Zurich, which is supervised by Professor Onur, who most of you should know. He has his bachelor's and master's degrees from the University of Kent on computer engineering. And his research spans bioinformatics, computer architecture topics, and I guess we're going to be seeing some of that. So,
Thanks a lot for the nice introduction. I hope you can hear me well. Yeah, seems to be the case. Then hello everyone. I'm Can. I'm a PhD student in the Safari Research Group at ETH Zurich. And today I'll present our work ApHMM, accelerating profile hidden Markov models for fast and energy efficient genome analysis. And I'm really glad that we too did some really nice background, introduction to genome analysis earlier today in the keynote. So let me start with an executive summary. So graph structures such as profile hidden Markov models are very commonly used to basically in genome analysis for accurate and fast analysis of biological sequences. But essentially, the parameters that are used in these graphs in profile hidden Markov models are mainly trained and used with a computational intensive algorithm known as the Baum-Welch algorithm. And this basically causes major performance and energy overheads for many genomics workloads. And our goal in this work is to enable rapid, power-efficient, and flexible use of profile hidden Markov models for genomics workloads. To this end, we propose ApHMM, the first flexible and hardware-software accelerator for PHMMs that can substantially reduce the unnecessary data storage, data movement, and computations by effectively co-designing hardware and software together. And we also provide a flexible design to support several genomics workloads that use profile hidden Markov models. And we basically do some extensive evaluation, and our ASIC implementation compared to some CPUs, GPU, and FPGA baselines across many different workloads shows that ApHMM provides significant speedups and also significant improvements in the energy consumption. So let me start with an executive with some background. So this work basically is mainly focusing on genome analysis, and you may ask why we do genome analysis, basically. And fast and accurate genome analysis is really important for many, let's say, applications. One is really useful for understanding genetic variations, species, and evolution. Another use case is it's really useful for predicting the presence of some harmful pathogens, let's say, in an environment. It's also very useful for a rapid surveillance of disease outbreaks, and also for, of course, personalized medicine. And basically, so how do we do this genome analysis, right? What is really the data here? So we have basically these genome sequencing machines that basically takes this biological molecule over there, which can be, let's say, a DNA or a protein. And what it really outputs is basically these small fragments of biological sequences. So I'm saying these are small fragments because they are really relatively much smaller compared to the size of your genome. So these can be in the length of thousands, while your entire genome can be in the length of 3 billion, let's say. And we have many of these fragments from the machines. And this genome analysis is really like when you have this data from the sequencing machines, this is really like solving a puzzle, let's say. So we really want to basically analyze sequences accurately and quickly, by comparing them. So you can assume that these DNA fragments are somewhat like these pieces of the puzzle, let's say, which can be biological sequences, let's say. So we really want to basically compare them to either each other to make some sense out of these pieces, or we may also want to compare to a certain template sequence, let's say, which can be a representative of a certain species or a certain group and so on. And this template sequence can be, for example, a human reference genome. And this is really essential. This sequence comparison is really essential to understand the functionality of a sequence, mutations, and diseases, and so on. And what we see is that the graphs are really commonly used in sequence comparison for two main benefits. So these graphs can really avoid redundant comparisons and storage. And they also provide rich information on, let's say, some expected variations or mutations between sequences or between individuals. For example, we may know that there may be some regions in your in the human genome that are not really prone to certain variations, but there may be some specific regions that is really prone to many mutations, and it can be really individual specific, let's say. And graphs can really store this information very effectively and efficiently. And profile hidden Markov models are basically powerful, and they are really common, basically, graph structures for sequence comparison. And the goal of these profile hidden Markov models is to identify variations between sequences probabilistically. So here, basically, in the graph, we, of course, have, let's say, states. And in PHMMs or in profile hidden Markov models, the goal is really when you visit a state, it outputs a certain character, or it emits, let's say, a certain character. And basically, when you see these characters, let's say, in a node, in a graph, you may say that, basically, this is the expected sequence. This is the sequence of DNA that we're expecting, let's say, in the most common case. And here we have ACTT. And basically, in order to basically output the entire sequence, of course, we need to visit all the states using some edges or transitions based on the observations, or based on the observed variations, basically, that we see in the sequence. So, of course, we have different types of variations. For example, if the sequence that we're observing is also ACTT, meaning there is really no difference between the expected sequence and ACTT, we simply basically follow this path and then somehow identify that there is really no variation in the sequence that we're observing. But of course, there are other types of variations, for example, substitutions. It may happen that compared to our expected sequence, we may have, let's say, slight substitution or variation. And in order to handle such cases, a corresponding state can somehow output a different character, let's say. And we have insertions. So we may have like inserted characters compared to the sequence that we're expecting. And there are also states that handle such cases. Here we have an insertion state, and when we visit it, it may basically output the insertion variations that we're observing. And similarly, if you have a deletion, let's say, compared to the sequence that we're expecting, we may simply skip the corresponding character and then somehow delete, or observe that we need to delete the character to identify that there's a deletion, let's say, deletion mutation over here. And of course, in order to support all types of variations at any position in the expected sequence, we have this particular pattern over here that allows insertions, substitutions, and deletions at any place in the expected sequence, let's say. And this is basically the structure of the profile hidden Markov model graph. And when you really look closely, what you really see is that there are essentially probabilities that are assigned to seeing each character in the DNA, and also probabilities assigned to visiting basically a state from another state. And these probabilities are really important in PHMMs, and to basically achieve accurate analysis. So in order to utilize these probabilities in profile hidden Markov models, we use a common algorithm known as the Baum-Welch algorithm for both inference and also the training of these parameters that you just saw, essentially. And the goal of inference is really to identify the variations between sequences, and the purpose of training is essential to maximize the parameters. The probability is to observe the certain variation, let's say, based on the observed sequence. And since this is somewhat ML session, I guess I'm also obliged to show you some, I guess, let's say, ugly equations, but really I'm not really going to go into the details of equations. But what we really have here is that we have, let's say, some equations that are called forward and backward calculations, and also some equations for updating the probabilities. And what you really see here is that we have somewhat like iterative nature of the computation. And this iterative computation is also somewhat dependent on the previous iterations. And the final calculation of these forward and backward values are used in the training step, in the later training step. So this is basically called training step. And also, this basically forward and backward calculation is really a dynamic programming approach, essentially. So in this calculation, what we really do is that we calculate the possibility of visiting each state in PHMM, right? So we have these states over here, given an observed sequence from both directions. Essentially, if our observed sequence is ATGT, in forward, what we do is that we process this character from start to end, and in the backward calculation, we process the sequence from back to forward, let's say. And this is why we have basically these arrows, these cells getting filled basically from this direction, and the backward, these cells are getting filled from this direction. And basically, once you calculate all the cells in this DP table, what you can do is twofold, is either inference or training. And if you really want to do inference, the goal of inference is, again, is to identify the variations between sequences. And what you can really do is that you can use the entire DP table and use some decoding algorithms to identify basically the states that are most likely to be visited, let's say, given an observed sequence using all these values in the DP table. And again, if you want to train your PHMM, again, you use this entire DP table and then use the equations that I showed you earlier to update the parameters, essentially. And PHMMs, since they are really powerful, they are commonly used in many genomics workloads, such as error correction, protein family search, and also multiple sequence alignment. But what we really see is that the Baum-Welch algorithm is essentially very costly algorithm. So when we look at these common workloads in genomics, so this is basically different workloads, and these show basically different steps in the Baum-Welch algorithm where rest of the, let's say, the percentage belongs to the other steps in that particular workload. But what we really see is that the Baum-Welch algorithm causes a major computational overhead in genomics workloads, taken up from 46% to the 99% of the overall execution time of the entire application. And this is basically because it contains some computational complex dynamic programming calculations, which are basically very compute intensive, including many floating point operations. And if you are basically familiar with hidden Markov models, you might have already noticed that the PHMMs are somewhat a specialized version of hidden Markov models with some fixed patterns, let's say, with some locality in the connections on states and transitions. And you might ask, basically, we could essentially use these HMM accelerators to also accelerate these type of applications. And what we really should see here is that when we look at basically the forward calculations in PHMMs, we have this nice basically pattern and, let's say, special locality in the calculations in PHMMs. But when it comes to HMMs, since there is really no restriction on the connections and so on, there is really no specific data pattern over there that we can exploit. So this is basically one problem. The generic HMM accelerators cannot really exploit the fixed data dependency pattern of PHMMs. We also basically want some flexible solutions to accelerate these PHMMs. And this is basically because PHMM requirements can change based on the application. So in some cases, we might use this design that I just showed you, or basically the design can slightly change while still providing all these functionalities of supporting insertions, deletions, and so on for sequence comparison. So that's basically one challenge that we may have. Another challenge is that we really want to basically analyze all types of biological data, including DNA and protein. So this means that the number of letters in our alphabet can also change, and the solution should really support that. But we see a lack of flexible mechanisms to handle different design choices. And also, there are many other problems. For example, there's we observe a suboptimal vectorization of SIMD-based solutions on CPUs and GPUs for many reasons, due to, for example, high warp divergence, branching, etc. And also, what we also observe is that a significant portion of these operations are really redundant for many reasons by some basically close understanding of the algorithms. So we also then observe existing solutions provide suboptimal solutions due to inefficient hardware and software design. So the problem is really then the Baum-Welch algorithm causes major performance overhead in important genomics applications, and hardware and software-only solutions are not sufficient for effectively accelerating PHMMs. So our goal in this work is to enable rapid, power-efficient, and flexible use of PHMMs when using the Baum-Welch algorithm. And to this end, we propose APHMM, which is the first flexible hardware-software co-designed acceleration framework that can significantly reduce the computational overhead of the underlying Baum-Welch algorithm for PHMMs. And we also provide a GPU implementation of our software optimizations, which is the first GPU implementation of the Baum-Welch algorithm, again, for PHMMs. We have essentially key software and hardware optimizations to accelerate this important application, let's say. And I'm going to start with describing the software optimizations that we propose. So our first software optimization aims to minimize the redundant data storage. And what we really observe here is that we don't really need to fill basically the entire backward table. It's essentially unnecessary to do so. And the reason is essentially for some pipelining opportunities that we observe in the equation, which allows us to directly consume the backward values in each cell while we calculate them. And basically, if you assume that we're using these particular cells once they are fully computed in the training, if you need to basically use this cell, then this means that you need this particular cell to be filled, and for this particular cell, you need this cell essentially. So there is that dependency pattern. But in this case, to use this particular cell in backward calculation, you only need this cell from the backwards direction, let's say. So then what we observe is essentially this part in the DP table is needed, although we don't really need to calculate this part in the DP table for backward calculation. So this means that this basically provides us some opportunity, essentially just only to fully calculate a single row in the DP table, and so that it can be used only in the next iteration of the DP calculation, which we call a partial compute approach over here. And we can do this basically for the entire table. And this is really useful for reducing the storage requirements during training. And the second software optimization that we're providing is basically to reduce the unnecessary computations in the Baum-Welch algorithm. And what we really observe is that there are certain cells in the DP table that are really negligible, that are not really affecting the end, let's say, result of the application. And our goal is basically to find the non-negligible states, the important states, let's say, or the important cells over here. And this is usually done by sorting. And the goal of sorting here is basically to find exactly, let's say, end cells or end states to basically find the largest values in the cell, because they seem to be the most important basically cells for the further calculation. However, sorting is, of course, usually very complex to implement in hardware, and it also includes some cost basically in it. And basically, the question is, can we filter without sorting? And the answer is somewhat yes. So instead of basically finding at least exactly end states or end cells, we revert the problem to finding at least end states, basically with the largest forward and backward values. And to this end, we propose this histogram-based filtering where the values in the cells are somewhat included in the buckets corresponding to their values. So if basically the value is large, they are inserted in the corresponding bucket, let's say. And what we can really do is that we can filter all these cells as soon as basically we find at least n states or n cells. For example, in this case, we may want basically to find 10 states. And to this end, we start with, let's say, the largest values, of course, and continuously ask whether the filter is full or not. So in this case, we have only 2 states. We continue searching until the filter is full. And when we basically notice that we included enough states for further calculations, we say, but the filter is full, so we can simply ignore the rest of the states. This basically reduces the computation a lot without having the complexity of sorting. And basically, the last software optimization is to avoid repeated operations. What we observe is that there are some multiplications that are redundant. This is mainly because of using the same default parameters or values for each possible connections in the PHMMs. And what this really causes is that we redundantly do some calculations over and over again that generates the same output. But to generate the same output, we perform these costly floating point operations to generate it, which is really costly, basically. And our goal is to avoid these redundant computations by enabling efficient reuse of common multiplication results by some memorization techniques. And to this end, we use basically cheap lookup tables, let's say, to provide these common results for us. So these were the software optimizations, and I'll quickly basically show you how our hardware design looks like, how we basically implement these software optimizations in hardware. So basically, this is the overview of our ApHMM design. We have 2, let's say, blocks in our accelerator, a control block and a compute block. The control block really receives the data from memory and also basically gets the essential commands from CPU to arrange the parameters that are going to be used, and also to understand what sort of data control is needed. And also we have our histogram filter implementation in the control block. And what it really does is that the proper indices are sent to compute block, and then we first calculate the forward value fully, because we need it to be computed fully. But once it's computed, what we can really do is that we can compute the backward values step by step. And while we compute the backward values, we can also start the training process. Again, as we compute the backward values, for example, we can do we can update the transition probabilities and also emission probabilities by somehow forwarding the backward values and also forward values to the next steps. And basically, this design is very flexible, and also it provides efficient control logic and hardware design that enables first opting out from our heuristics, such as lookup table and filtering and so on, because some applications may really need really accurate computation. And it also provides the necessary controls to give the right, let's say, index control for different design of PHMMs. And if you basically look more closely to the compute block, what it really does is that it performs the forward, backward values and updating the transition values in one large, let's say, module, and also updating the emission probabilities are done in another module over here. And here we have several processing element groups. Each of them is including 4 processing elements, and each of them is responsible for calculating forward, backward values and also updating the transitions. So what we really have is that these, let's say, units that can do the computation accurately and receive the data off chip. Here the red arrows show the off chip accesses. So it starts with accessing the right data that needs to be used in the current iteration, and then the right output can either be outputted to the off-chip memory, or it is essentially broadcasted to the next step, which is it is streamed basically to the next step, which is updating the transitions and so on. And similarly, we stream the data for updating the emission probabilities and so on. So basically, we efficiently exploit some data locality, broadcasting techniques, memorization, streaming, and pipelining, and so on, with our software optimizations for an effective hardware-software co-design, which provides us significant benefits, which I'm going to essentially quickly discuss our results now. So in our evaluation methodology, we evaluate performance, area, and power. We have a synthesized model using a 28 nanometer processing technology at 1 gigahertz. Our CPU baseline is a high-end CPU using a single thread, 12 threads, and 30 threads for many applications. Our GPU baseline also includes somewhat a mid-range and also high-end GPUs, and we have also FPGA baseline based on an earlier state-of-the-art work. And our use cases are, we have 3 use cases that supports PHMM design, and also their software, state-of-the-art software baselines. We have error correction, protein family search, and multiple sequence alignment. And our comparison points are, again, to the state-of-the-arts. For CPU, we compare against Apollo and HMMR. For GPU, we compare against, again, our GPU baseline, GPU implementation, also the state-of-the-art accelerator for hidden Markov models, and also to the FPGA state-of-the-art work. And our data sets are from real world data sets. We use around 10,000 real DNA sequences for error correction. And we use basically thousands of PHMM graphs and, again, thousands and millions of protein sequences to perform really costly protein sequence similarity search for the important workloads. So I'm first going to show you the acceleration of the main kernel, which is the Baum-Welch algorithm over here. This figure shows the speed up that each optimization or each basically implementation achieves over the single threaded CPU implementation. And we have basically each step in the Baum-Welch calculation over here, forward calculation, backward, and parameter update step. And this is basically the entire Baum-Welch algorithm. So what we really see is that APHMM provides significant speed ups up to 260x, depending on essentially the baseline, of course, compared to CPU, GPU, and FPGA implementations of this particular kernel. And what we also see is that for forward calculation specifically, GPUs provide essentially better performance. And what we realize is that this is mainly due to the frequent off-chip memory accesses that ApHMM makes because we calculate the DP table for forward fully. And we also look at the workload acceleration and similar figure, speed up over CPU, single-threaded CPU. We have different workloads over here and using multi-threaded and also different baselines here. So again, what we see is that for end-to-end acceleration of these workloads, specifically even for error correction, because it really uses, let's say, the training a lot, for error correction, we achieve significant speed up for the end-to-end acceleration of the workload. And also for other workloads, since it constitutes around 50% of these workloads, we can also achieve somewhat up to basically almost 2x over there for accelerating these workloads. And when we look at, again, the energy reduction as well, significant, again, energy reductions with efficient, let's say, hardware-software co-design of these PHMMs. And again, for the workloads, when we look at these end-to-end reduction of the energy for the workloads, again, significant reduction over here. And we also show the speed up of each optimization that we're making. And what we really see is that I really want to focus on this third optimization here, which is basically somewhat the combination of broadcasting and partial compute. So the partial compute is a pure software optimization, while broadcasting is really implemented in the hardware. And this is the biggest speed up that we're obtaining in the design. And what we really see is that this broadcasting and partial compute together is only possible with an efficient hardware-software co-design. And we also analyze the area and power, and overall, the area and power requirements are relatively small, but the UT unit over there, updating the transition unit, requires the largest area due to several complex units, such as multiplexer, division pipeline, and local memory, and so on. So we have more results in the paper. If you are interested in learning more, you can check these, and we also have more details about our mechanism and the configuration and so on. This paper was published in ACM TACO very recently, and this is it's a collaborative work with the Intel folks at TU Delft and Carnegie Mellon, and of course that's ETH. Our GPU implementation is fully available in our GitHub page, so this is the QR code if you want to check. And to conclude, our goal is to accelerate PHMMs, accelerate Baum-Welch algorithm for PHMMs, and we provide the first flexible and hardware-software co-design that can do that, and we show significant speedups in terms of and also energy reductions by doing so. With that, I'll conclude my talk, and I'm happy to take questions if there are any. Thanks.
Okay. Yeah. Thank you, Chan. Those are wonderful. Any questions for Chan? Sure.
Thank you for your presentation. I have a question about, you have said in order to reduce the computation, you're using not meaningful, you use something to find the states. So how the hyperparameter state is defined and how to choose the hyperparameter n. You just tell us if you decided in n how to choose the n states, but how is n is defined and how to choose n, and how is the n influence your results? Thank you.
Really good question. So we did an empirical analysis for that. We identified basically how many n's that we should pick in order to achieve almost identical accuracy that we can achieve without filtering. So this is really based on some empirical analysis. And of course, here we show both the effect of n in terms of both speed up and also accuracy. What we really see is that in order to achieve high accuracy, of course, we need n like somewhere around here. But of course, there is also a significant drop in let's say, the time requirement as the n increases. So there is somewhat a sweet spot over here, and this is. Does that answer your question?
Oh, okay. Mostly on the experiments or the results, we need to do the grid search for the n.
So we don't really do, let's say, these, I guess, ML focused grid searches to identify the parameters for this and at least. The reason is they really need basically to run the entire application, and this application is pretty slow essentially. If we really need to do some research over there, I guess it will take really lots of time, I would say. So we basically predefine certain parameters over here, starting from some value to another, and then run the application basically several times. In this case, I guess we run it maybe 10 times for each parameter, and then basically see what is the motivation here, depending on the time.
Thanks. Any other question? So yeah, I do have one. So you guys have done quite some work also on near memory computing, right? And 3D stack DRAM. So I was surprised that this is not near memory, especially because you mentioned that the data movement was one of the reasons why in some cases the GPU was faster than the accelerator. Can you comment on that?
So I guess, yeah, that's a good question, and also, but I guess relatively hard to answer. So in our case, at least for the applications that we looked at, what we saw is that the computation was really the issue over here. So that's why we focused on the ASIC implementation here. But again, we did also an analysis, how many cores, how many accelerators of ApHMM we can use without really being bottlenecked by the data. And what we see is that there's a limit to it. In this case, we can use around 4 cores, 4 accelerators, and if we basically go above 4 cores, what we see is that the overhead is basically causing a slowdown compared to the best result that we're getting with the 4 accelerators. Although the applications that we've chosen basically fits really well with the 4 accelerators because we can achieve significant speed ups and so on, but we still argue that there might be other applications that may need even more accelerators, a larger number of accelerators, but can basically show slowdown with this market overhead. And if we can find such applications, I think there's an opportunity here to minimize that data movement overhead with processing memory or processing near memory approaches. But currently we're lacking that motivation for applying this for different applications. And I would be really happy basically if we can find such an application, suitable application where we can exploit it.
Good, thanks. Maybe one last question that I want to get rid of. So you mentioned that your design is flexible. You can parameterize some things, right? Like for instance, you can play with the alphabet, you can change the alphabet, you can. So how is this? If I'm a user and I have a use case, how should I interact with this accelerator? How to access those configurable parameters, or this is just design time parameters?
So I'm not sure. So is the question how the end user defines these constraints in the design with different alphabet sizes?
Or if I have a design, if the ASIC is my system, and I want to use it, but for a slightly different application, maybe with different alphabet, how should I go about doing that?
So that's a good question. I guess I'm assuming you were thinking of, increasing the number of letters, combination of them for different applications. We have somewhat an empirical analysis in the paper, although I don't have those slides over here. We also discussed these non-genomics workloads and how we can support it. Although being flexible is important here, it also causes, of course, extra overhead, right? So the area and the power overhead of this is also caused by this flexibility. And I guess if the alphabet size increases, your on-chip memory size also needs to increase, which will increase, it will increase almost exponentially, basically. So this is the trend that we're seeing. And it will also increase the power and area requirements. I think there's a challenge in supporting even larger number of letters and so on for different applications, but I think that's also a good research direction, how to make it even more efficient and, I guess, fast.
Okay, cool. Yeah, let's thank the speaker again.